also some where you could see partially fragmented. And if he did a cell count, he observed that in response to 100 nanomolar rotenone, there was a large increase in the percentage of the neurons that had fragmented mitochondria. Zung also did some gene expression analysis for OPA1, which is a gene that's involved in, again, the morphology, the dynamic morphology of mitochondria and in apoptosis. And he saw that 5 days post-fertilization in fish that were treated with rotenone, the expression of OPA1 was reduced compared to control. This is another gene that's involved in mitochondrial dynamic, but this one didn't seem to change that much. However, a few days after the treatment had been interrupted, the fish seemed to be able to react by re-increasing the expression of OPA1 compared to control. So again, just an example of the use of these transgenic fish with fluorescent markers that label either the dopaminergic neurons or the mitochondria within dopaminergic neurons. Okay, and the last part of today's talk, the longest part, I'm going to talk about chemogenetic ablation of the dopaminergic neurons. And that's work that was done by Raphael Godoy, a former PhD student in the lab. Julia worked closely with Raphael when she was in Ottawa. And this work is continued by Kang Hua, a current PhD student in the laboratory. So this chemogenetic ablation relies on the transgenic expression of a gene coding for an enzyme called nitroreductase. Nitroreductase is originally from bacteria. And the expression of nitroreductase in an eukaryote or in cells of an eukaryote has by itself no effect on the function of that cell. Okay? However, if this eukaryote is exposed to a prodrug called metronidazole, which by itself doesn't have much of an effect on the organism, and that depends on the dose that you're using, but at some doses, metronidazole by itself has no effect. Well, in the presence of nitroreductase, metronidazole is turned into a cytotoxic compound that will cross-link DNA and cause cell death. But it will only cause cell death in those cells that make nitroreductase. So the beauty of this system is that you can control where and when death will happen. You control where by controlling in which cells the nitroreductase is made, and you control when by controlling when you administer the prodrug metronidazole. So just to take the DAT transporter will target nitroreductase to the dopamine neurons, but that starts, in late embryos, early larvae. But you can control when the cells die, because if you wait until they're adults to administer the metronidazole, then the death of the dopaminergic neurons will happen only then. Okay, so to target this, we use this CFP, cyan fluorescent protein, blue, with nitroreductase. We use exactly the same approach to insert this fusion protein in frame with the first exon of the DAT transporter gene, just like we did for EGFP. And then Raphael, the first thing he did is to see if there was a decrease in the CFP expressing cells, dopaminergic neurons, or using that as a probe. Okay, so here on the top line you have the DMSO control. So that's we dissolved metronidazole in DMSO, and in the presence of metronidazole, there is a decrease in the number of DAT expressing or CFP expressing cells. The panels on the right are just the same kind of experiments but carried out in the EGFP fish, so it's some sort of a control to show that metronidazole by itself does not have any impact on the dopaminergic neurons. So Raphael then did a more detailed examination of what happens in those fish. So he looked at various regions of the brain. So here in this lane are the ventral diencephalic cells, so 2 clusters of these cells in the diencephalon. Here you have also the olfactory bulb. I point your attention to them because we're going to talk about them a bit later on. And he saw that decreases were observed in the presence of metronidazole, so chemogenetic ablation was obtained in these various brain regions. He also saw that chemogenetic ablation was paralleled by an increase in the expression of active caspase 3, which is involved in the death pathway. And here we see the controls, the CFP expression, this is just DAPI for the nuclei, and an increase in caspase 3 expression in those fish that receive the metronidazole. In collaborations with colleagues at Carleton University, we also used HPLC to look at dopamine levels following ablation. So shortly after treatment with metronidazole, there's a decrease in dopamine levels, but then if you let the fish recover, the dopamine levels go back to normal afterwards, which is kind of indicative that the fish is able to compensate, presumably by making new dopaminergic neurons once you have stopped administering the metronidazole to the fish. Raphael also saw that there was some phenotypic consequences in ablating the cells. So for instance, if you look at the locomotion of the fish, you have the controls in blue and the metronidazole treated in red. Shortly after treatment, there is a decrease in total locomotion in the treated fish compared to the controls. But again, if you remove the metronidazole a few days later, this difference is not observed anymore. Okay? And you can, observe locomotor control in a number of ways. You can look at the angle that the fish larva takes when it initiates movement. You can look at a number of different parameters. The beauty of this is all this can be done in an automated way. And Raphael examined the changes in these parameters in great detail. But the big question we were asking ourselves in these experiments that are done during the development of the fish is whether this response of the fish was due to an increased production of dopaminergic neurons once you stopped ablating them with the metronidazole. And that answer in the experiment that Raphael did was not immediately obvious. Okay, so he looked using bromodioxyuridine labeling. So it's a reagent that allows you to see increased proliferation, let's say, or cell division, and DNA synthesis, then cell division. So by BRDU labeling and looking at the the presence of BRDU, indicative of cell division, and CFP, which I remind you is expressed in these transgenic fish in the dopaminergic neurons. So the cells that are double labeled for CFP and BRDU are newly made dopaminergic neurons. But as you can see on the right, if you compare the vehicle with the metronidazole treated fish, there was a little bit of an increase, but this increase did not reach statistical significance. So one simple way of interpreting these data would be to say that at the time this experiment was done, the fish made so many dopaminergic neurons anyway that in response to there was a loss of these dopaminergic neurons, but it did not, I'm kind of using words that the fish don't think that way, but the fish didn't feel it was necessary to increase the production to compensate for the neurons that we had been killing at the time. And you'll see other examples in adults that kind of indicate things are a little different there. Okay, so now we're going to go from embryos and early larvae to the adult zebrafish brain. So I mentioned earlier that fish have this ability to regenerate a lot of things after injury, but fish are also different from mammals in the sense that their ability to create new neurons during adult life is a lot higher. Okay? In the mouse, we generally consider that there are two neurogenic zones where new neurons are being made in the adult mouse brain. And in the zebrafish, work by Michael Braun's lab and other labs have shown that there's up to 16 of these neurogenic zones, and these are distributed pretty much all around the brain. Okay, so in a simple way, we can say that the brain of the zebrafish sees the generation of new neurons throughout the life of the animal. So that's your entire brain. But again, just to locate things a little bit, these are the dopaminergic neuron regions, those that are labeled with the CFP nitroreductase transgene, okay, in a section, a sagittal section of the adult brain. And this big circle over here points to the olfactory bulb. And for reasons that are not absolutely clear at this point, we saw effects that were a lot stronger in the olfactory bulb compared to the other parts of the brain. I'll try to remember to mention them. But a lot of data that you will be seeing now are from the olfactory bulb of the fish. So just to give you an idea, okay, so this is the olfactory bulb, and then you have, CFP or the expression of the DAT gene, etc. So there's a lot of things, there are a lot of cells over there. So Raphael and undergraduate student Victoria Marie Cusson did some experiments. So they treated the fish for 1 day with 10 millimolar metronidazole, and then they observed the brain at various time points. And here are 3 sections that are made through the olfactory bulb. And in response to metronidazole administration, you see these decreases in the number of CFP positive cells, therefore in the number of dopaminergic neurons. Again, using HPLC, we observed transient, so temporary decrease in dopamine levels following the metronidazole treatment in the brain of the adult fish. Again, in the olfactory bulb, but also, in the ventral diencephalon, hindbrain, etc. Okay. But then, current PhD student Kang Hua was interested in looking at same question Raphael has asked: does the fish make new neurons in response to the ablation? And if the fish makes new neurons in the ablation, when does it do that? And in this experiment, he used 2 methods to label newly born cells: the BRDU that I mentioned to you earlier, and EDU, which is another of these DNA labeling agents. Okay? And I forgot to put that here, but the way the experiment was done is that the cells were ablated with metronidazole. Then he administered BRDU for a few days to look at the neurons that are made during these few days after the end of the metronidazole treatment. And then he administered a large dose of EDU or larger dose of EDU later on to look at those neurons that are made after, so several days, let's say, after the end of the metronidazole treatment. And I think the results over here, and here there are results, again, labeling in the olfactory bulb. And again, as we did before, Kang counts the number of cells where BRDU is present, so that is, new cells that are made, presumably neurons, but also cells that are double positive for BRDU, newly made cells, and CFP, newly made, or dopaminergic neurons. So there are 2 kinds of data. So if you look here in the top part, okay, these are the total numbers of BRDU positive cells, the new neurons. Unfortunately, Kang did that using percentages, but here, if we look at counts, we're talking about, let's say, in a particular region, at about, let's say, 6,000 cells. Okay? And there's no difference between the controls and the fish that had undergone chemoablation with metronidazole. However, if you look at the cells that are double positive for BRDU and CFP, then we see an increase. And that's because these cells, instead of being, let's say, 6,000 of them, we're talking about, let's say, between 50 and 60 cells. So the cells that are double labeled for BRDU and CFP represent a very small percentage of the total cells that are labeled with BRDU. In other words, in simple terms, the fish makes a lot of new neurons in adults, okay, regardless of whether you kill them with, you administer metronidazole or not, but only a small percentage of these neurons that are being made are dopaminergic neurons. So you really need to look at double labeling in order to be seeing anything. So the top part of the slide is the BRDU labeling. BRDU was administered at the end of the chemogenetic ablation. Then if you administer EDU later on, well, again, same kind of thing. There's a large number of cells that are EDU positive. But then when we look at the cells that are both positive for EDU and CFP, dopamine marker, then there's not much of a difference. So what this says, what this suggests, is that the response of the fish to increase the synthesis of new dopaminergic neurons after chemogenetic ablation occurs in the first few days, but doesn't seem to be occurring later on. Maybe later on, just the normal synthesis of, or the normal generation of new neurons is sufficient. Okay, Kang also looked at a number, using digital droplet PCR, looked at the expression of a number of genes at several time points after the treatment with metronidazole. And if you look at the expression of GFAP, which is an indicator of glial cells or radial glial cells, these cells that participate in the generation of new neurons in the adult fish brain, or nestin, another indicator of early cells, early newly made cells, there is an increase in the expression of these genes after metronidazole treatment. SOX2 is another indicator. And at that stage, Kang also looked at the expression of genes in a number of pathways, signaling pathways important, in nervous system. For instance, the Wnt pathway, the sonic hedgehog pathway, or the Notch pathway.